Hello dear viewers, myself Nasir and you're watching Mr.Zoologist YouTube channel. Today we are going to discuss about biomolecules. What are biomolecules? They are in actual chemical compounds that are found in living organisms. Bio means living and molecules means molecules. So they include organic and inorganic compounds that may be made up of both carbon and hydrogen or may be made up of only carbon or only hydrogen. These are called as biomolecules. Analysis of a chemical composition in a tissue, grind a living tissue like vegetable or piece of liver in trichloroacetic acid Cl3COOH to get a thick slurry. Slurry means it's powder or paste. Strain this through a cheesecloth or cotton to get two fractions such as filtrate and the retinate. Retinate means to say its insolubilities and filtrate is soluble. And filtrate, what is filtrate or acid soluble pool? Contains biomolecules. Molecular weight is less than 1000 dalton, whereas retinate or acid insoluble fraction contains macromolecules which have molecular weight greater than 1000 dalton. Analysis of inorganic compounds, weigh a living tissue wet weight and it is dry weight to evaporate water, to means to say remove all the water, dehydrate it. It is fully burnt to oxidize, means to say add oxygen. All carbon compounds to gases form like carbon dioxide and water vapor. It forms ash. Ash means all the compound is burnt totally. The ash contains inorganic elements like calcium, magnesium, sodium, potassium, etc. and inorganic compounds like SO4 2 negative, PO4 3 negative, NaCl, CaCO3, etc. Comparison of elements in non-living living matter: hydrogen 0.14 in earth crust, human body 0.5, carbon 0.03, 18.5 in human body, oxygen 46.6 in earth crust, in human body 65, nitrogen very little in earth crust, in human body 3.3, sulfur 0.03 in earth crust, 0.3 in human body, sodium 2.8 in earth crust, 0.2 in human body, calcium 3.6 in earth crust and in human body 1.5, magnesium 2.1 in earth crust and 0.1 in human body, whereas silicon 27.7% in earth crust and negligible in human body. Biomicromolecules, molecular weight of biomicromolecules found in the acid soluble pool ranges from 18 to 800 daltons. The acid soluble pool represents a cytoplasmic composition, means to say include amino acids, lipids, sugars and nitrogenous bases. What are amino acids? They are the compounds formed of an amino acid group or an acidic group. Amino group and an acidic group COOH, NH2 and COOH. That NH2 and COOH are attached to the same carbon atom, that is called alpha carbon. To whom all the functional groups attached, that element is always called as alpha. To the next to it is called beta and up next is gamma and so on. They are called alpha amino acids. They are substitute methanes, means to say they are derivatives of alkanes. If we replace R or alkyl group with the help of hydrogen, that amino acid will be called as glycine. If we replace alkyl group R with that of CH3, that will be called as alanine. If we replace alkyl group R with CH2OH, that will be called as serine. Out of 20 types of amino acids that are used in protein synthesis, amino acids are of three types. Acidic amino acids, which contains an acidic group, that is carboxylic group. Basic amino acids contains an amino group, that is why they are called as basic amino acids. For example, lysine. Neutral amino acids contain both of them, is acidic as well as basic group, so they are called as neutral. They are called, for example, valine. Some amino acids are aromatic like tyrosine, phenylalanine and tryptophan. Out of 20 amino acids, 10 are essential and 10 are non-essential. Essential elements cannot be synthesized by the body, should be supplied outside from the body through diet, like lysine, leucine, isoleucine, tryptophan. Non-essential amino acids are prepared by the body, do not require from outside, like glycine, alanine, serine and arginine. In amino acids, amino and carboxylic group have ionizable nature, so structure of amino acids changes in solution of different pH. If both amino and carboxylic group ionize, it is called as Witter ion, because H from the carboxylic group moves towards the amino group H2N or NH2, where it forms NH3 and in the next process both hydrogen from the NH2 that comes from the carboxylic group and one from the amino extra ammonium ion group that will move in the form of hydrogen gas outside into the environment. Lipids are organic compounds, biomolecules, which are water insoluble, contains carbon, hydrogen, and oxygen. But number of oxygen atom is less in lipids. Lipids store double amount of energy as that of carbohydrates. Lipids can be simple, can be compound, or can be derived. If we talk about simple lipids, they are formed from fatty acids and alcohol like glycerol. Fatty acids are lipids with hydrocarbon chain R group ending in COOH group, that is called simple lipids. For example, palmitic acid has 16 carbons, whereas arachidonic acid has 20 carbons. As you can see in the diagrams, these are the structural formulas of these fatty acids. If we talk about types of fatty acids, are of two types: saturated fatty acids and unsaturated fatty acids. Saturated have no double or triple bonds between carbon atoms. For example, palmitic acid and stearic acid. Palmitic acid contains 15 carbon, whereas stearic acid contains 17 carbon atoms. And you can see in the diagram of stearic acid there is no double bond in the whole structure between carbon to carbon. We are not talking about between different atoms. We are talking about carbon to carbon. There is no double bond in the whole chain of the saturated fatty acids. Whereas if we talk about unsaturated fatty acids, they have one or more carbon to carbon double bonds. For example, oleic acid, arachidonic acid. As you can see in the diagram of oleic acid, there are multiple carbon to carbon double bonds, but here we are looking at the oleic acid, so it has one double bond. If we talk about the structure of the glycerol or trihydroxypropane, three carbons are attached with the OH groups, hydroxyl groups, and three have hydroxyl groups. That is why they are called as glycerol or trihydroxypropane. Fatty acids are esterified with glycerol through ester bond forming monoglycerides. If two combines, that will be mono. If single combines, that will be mono. Two combines, diglycerides. Three combines, triglycerides. One hydroxyl group from one glycerol and one fatty acid will form a lipid. If one glycerol plus one fatty acid, mono. If one glycerol plus two fatty acid, that will be di. If one glycerol plus three fatty acid, that will be triglyceride. As you can see, monoglyceride next towards the downside, and then the diglyceride that contains two hydroxyl groups, whereas two esters groups, these will be, if it contains three esters group, that will be triglyceride. So that is the structure of the fatty acids. In triglycerides, we have three alkyl groups, and in actual three hydroxyl groups are forming on the chains of the alcohols. Fats have higher melting points, whereas oils have lower melting points. So that is a major difference between fats and oils. Fats are solid, whereas oils are maybe solid or liquid at room temperature. Next is compound lipids. These are esters of fatty acids and alcohol with additional groups. For example, phospholipids, it is a combination of fatty acid plus glycerol plus phosphate. They are found in cell membranes. They are made of cell membranes, like lecithin is on the left side, you can see in structure, and on the right side they are in actual lipids are insoluble in water, so they form the structural components of cells.